Hello everyone and welcome to Pocket Size Bio, Dr. Casco. Today's video we're going to continue the Campbell Biology textbook with Chapter 7, Membrane Structure and Function. This chapter is broken down into 5 sections. In this video we're going to go over section 7.1 and 7.2. Talking about cell membranes, our fluid mosaics of lipids and proteins, and membrane structure results in selective permeability. So let's get to it. Zooming into the chapter image that you see here, figure 7.1, successful learning relies on communication between brain cells. Here, the vesicles fusing with the plasma membrane of the top of the cell releases molecules that are in yellow. So if you see this top cell here, you see these vesicles that are kind of flowing to the border, to the cell membrane. Inside those vesicles, you have these yellow small little molecules. And these yellow molecules, they bind to membrane proteins. Okay? And these membrane proteins are in light green that you can see on the edges protruding out of the cell membrane there. And particularly, they're protruding out of the bottom cell. And they're going to trigger these proteins that are going to act, that are receptors, to change shape. That change shape leads to a sequence of biochemical pathways that will enable the whole nerve cell and brain to essentially trigger learning. Okay, the plasma membrane that surrounds each cell regulates its exchange with its environment and surrounding cells. So we're going to look at what exactly happens and how it happens in this system of transportation of material. So how does the plasma membrane regulate inbound and outbound traffic? We're going to take a look at different ways: bulk transport, passive transport, active transport. And then within these different ways, you have more specific carrier proteins and transport proteins and channel proteins. You have exocytosis and endocytosis. Okay. And you have active transport, where you're going against the gradient. Material is going against a concentration gradient. So let's move to 7.1. So 7.1, you have cellular membranes are fluid mosaics of lipids and proteins. Just to move a little bit down here. As we know already, right, the cell membrane, it's made up of a phospholipid bilayer. Okay, but the membrane itself of cells, they have the phospholipid bilayer, and they have proteins that are embedded within that bilayer. Okay, we need to really understand the idea of phospholipids. So we're going to look at the structure really quick. These are lipids that are amphipathic, meaning that they have regions that are hydrophobic, water-fearing, and regions that are hydrophilic. Because of this property, they form a phospholipid bilayer, which creates the boundary of the cell membrane. And it can divide the cell's internal contents with the extracellular environment. Okay, so the hydrophobic area is referred to as a hydrophobic tail, or tails. Okay, and you have the hydrophilic heads. If you guys remember, we have that phosphate area, and you have the hydrophobic, basically the hydrocarbons, right? That sometimes they have kinks and whatnot. So you can see a molecular structure of a representation here. Again, so they're amphipathic. The hydrophilic area will protrude out of the membrane, right? Exposed to water, right? To the internal contents or the external environment. And then you have the hydrophobic areas, and this would be considered a non-aqueous environment, right? And they're housed in between the bilayer. Okay, so the cell membrane is um, described as a fluid mosaic model, okay, because of um, the nature in which the membrane is constructed. You have proteins that are embedded within the cell membrane, and it's referred to as the mosaic of this model. Okay, this is the currently accepted model that scientists use and on which all the molecules are arranged in. And these proteins, they're floating in a sea or in a fluid of phospholipids. And it's a very, very dynamic structure. It's referred to as a mosaic, just like certain artwork in which you would embed pieces of, various materials or various designs, into artwork. Proteins are embedded in that way within the phospholipid bilayer. Okay. As we're going to see later on, proteins can be found into specialized groups. You can have several proteins kind of patched together or clumped together for a common function. It could be for enzyme activity, where the proteins can break down a molecule into a product 1 and then go into a product 2. Enzyme 2 will produce product 2, and so forth, like a relay system. Okay. Lipids have been found to have this behavior also working in groups or in patches, so much so that they're called lipid rafts. How much this actually is true is still, being studied. Some scientists say, if these structures are actually present and have an actual function, or it's some biochemical artifact, right? Or byproduct of some staining technique or whatnot. Okay, so, but they're there, right? They're referred to as lipid rafts. So we're going to look at the cell membrane more in detail in this chapter, right? We've seen how proteins can be embedded on this cell membrane. Some of these proteins can protrude throughout the entire membrane, transmembrane proteins, or you can have some peripheral proteins that are at one side of the cell membrane. Aside from that, you can have carbohydrates, which would function for ID tagging and so forth. So we're going to see all these more in detail. Let's continue on. Again, so going into the phospholipids a little bit more in detail. Within the aspect of fluidity, this is a very important concept for membranes. Okay, so phospholipids, this whole bilayer, is not a static structure. It's very dynamic. It's very fluid in the sense that molecules are moving about. The cell, the phospholipids themselves, they kind of move from position from one place to another, and even the proteins could be moving and kind of floating, let's say like a boat or a raft, just moving because of the dynamic nature of the phospholipids. So it's very dynamic. And of course, these phospholipids and all the molecules, they're held by hydrophobic interactions. This is what I was just mentioning here in the notes, that most proteins and lipids, they shift sideways. So they can move along with the slide past through each other, move along each other. Each molecule of a phospholipid, right, is passing through each other like a crowd, right, that is constantly, displacing each person there. And it moves pretty fast, right? The individual phospholipids within the membrane, they can move and exchange their location with others. They can switch 10 to the 7 times per second. Sorry about that little annotation there. I couldn't find the little power there. So 10 to the 7 times per second. And they can move 2 micrometers per second. That's how fast they can be moving. Okay? The phospholipids. Proteins being a lot larger, they can move, but they're slower. Okay, just because of the size there. Some of them can be immobile, meaning that they don't move because they're latched on to the extracellular matrix or just to some fibrous proteins there. So they're relatively stagnant. Okay. So Also, they could move in a highly directed manner because of the skeletal fibers, like we've seen in the previous chapter of motor proteins. Okay? Now, here's probably one of the more important things that you need to know. Membrane remains fluid as temperature decreases until the phospholipids settle in a tightly packed arrangement. So the idea of fluidity in the cell membrane is very important. You cannot have a membrane being too fluid, or else material is just going to move in and out in a very unregulated form. And it can't be too packed either, or else material won't come in and out. Okay? So it's very important for a cell to be able to regulate this level of fluidity. Okay? And the way that it does that is by cholesterol. Okay, so we're going to get into it in a little bit. So remember when we talked about lipids and the idea of unsaturated and unsaturated lipids being able to be a solid at a certain state or a fluid in a certain state. So specific temperature for to solidify depends on the type of lipids that it has and if it's unsaturated or not. So if a membrane is rich in phospholipid that are unsaturated or that they have unsaturated hydrocarbon tails, could determine how fluid they are. The more kinks you have in these tails, right, the membrane will become more fluid, okay? And that will prevent tight packing. So the more kinks you have, the more spaces you have, essentially, it means you can be more fluid. And what does that mean? It means that the phospholipids can move about amongst each other. They can let material pass through within those little spaces. So hence the word fluidity, acting more like a liquid. Okay, and then the more saturated tails that you have, the membrane will become more viscous, meaning that they're more tightly packed, so they have a more solid-like texture that will prevent material from moving comfortably throughout the membrane. So you want to have a good balance there for the cell to receive its material effectively. So again, what happens here is that you have a molecule, you have a lipid, that is cholesterol. Cholesterol is very, very important for the cell membrane. It is not a bad thing for the body. You need cholesterol to regulate that fluidity. So cholesterol is wedged between the phospholipid molecules, and it can have different effects on the membrane fluidity depending on the temperature. So take a look at this diagram here. This is essentially how cholesterol looks like. Okay, it's wedged in throughout the cell membrane, and this goes all throughout, right, the cell membrane. Okay, so when temperatures are relatively high, so you have a high temperature like 37 degrees Celsius, it's close to our body temperature, cholesterol makes the membrane less fluid. Remember, the higher the temperature goes, molecular motion starts to get faster and faster and faster. We don't want that. Or it's going to be too fluid, it's going to be too leaky. So cholesterol helps maintain and restrict that fluidity. Okay, it keeps the phospholipid molecules from moving too much. So it kind of binds them together a little bit, okay, blocking their motion. At the same time, at lower temperatures, cholesterol decreases, okay, the temperature that you need for solidification, for tight packing. So let's say you go down to 15 degrees Celsius and then you get a lot of packing. But if you add cholesterol molecules, you can lower it down to 10 degrees Celsius, okay, just to give a sense of what I'm referring to as decreasing the temperature. So if you can decrease the temperature with cholesterol, it can remain a fluid for better, okay? Having cholesterol. Without cholesterol, then everything will be packed very tightly. Okay? So cholesterol reduces membrane fluidity at moderate temperatures. okay, by reducing phospholipid movement. But at low temperature, it hinders solidification by disrupting the regular packing of phospholipids. How can you tightly pack if there's something in the way from doing that? So ultimately, cholesterol serves as a fluidity buffer. Okay, membranes must be fluid to work properly, as I mentioned before. This helps affect permeability, and this also helps protein movement. Okay. So again, similar to olive oil in a sense, right? To be fluid. So if the membrane solidifies, membrane enzymes can become inactive just because they can't move, right? And it's difficult to interact with other molecules. If the membrane is too fluid, interactions cannot occur also because there's just the rate of movement is just too high. It's going to impede in actual interactions. Okay? So you want to avoid the extreme environments in this sense, and that's what cholesterol helps the cell membrane achieve, right? Avoiding the extremes. Okay, because of this idea of cholesterol and fluidity of the cell membrane, certain organisms have evolved to be able to thrive in different extreme environments, if I can say, just because of that change in lipid composition, right? So variations in the cell membrane exist, right, through many species, and this serves as evolutionary adaptations to maintain appropriate fluidity. Again, always under certain conditions. The environmental conditions will kind of define the lipid composition. Okay, some examples is that you have this Antarctic ice fish. Okay, they live in extremely cold environments. So the way that they are able to do that is because they have a high proportion of unsaturated hydrocarbon tails. Remember, if the more hydrocarbon tails you have, plus cholesterol, this will enable the membrane to be fluid at a very low temperature. So it could still be fluid. So that will prevent packing of the phospholipid bilayer. Okay. Some archaea, you have these extremophiles, right? If you remember from high school biology, that thrive at 90 degrees Celsius, incredibly high temperatures in these hot springs or these geysers. And they have very particular lipids that they prevent excessive fluidity, right, at high temperatures. So they're more tightly packed together, preventing fluidity at relatively high temperatures. Thermophils. Okay, so the idea of changing lipid composition has enabled organisms to thrive in different environments. Very interestingly, you get there's a plant called a winter wheat that can still be able to thrive in the winter, right? It could tolerate extreme cold just because the percentage of unsaturated phospholipids increases in autumn. Okay, increases in autumn. So ultimately, that will prevent freezing during the winter. So this is a picture of the winter wheat, in the early stages of growth. Okay, and also other bacteria and archaea have this ability, okay, to thrive in these extreme environments. So ultimately, natural selection favors organisms who can get a mix of membrane lipids to be able to thrive to these particular conditions and be able to regulate it rather effectively, right? Okay. So talking about that phospholipid, now we move on to the membrane proteins, okay? The mosaic part of this fluid mosaic model. So this is the mosaic aspect, and the membrane is a collage of different proteins. So you have a lot of proteins that are embedded within the cell membrane. Again, as I mentioned before, they can be clustered into groups depending on the function that they need to do. Okay, and the amount could vary depending on the cell type. You can have 50 kinds of proteins on red blood cells only, right? So ultimately, what you need to know is that the proteins that you find in the cell membrane can help determine what the membrane does and the overall cell does. Okay, so different cells have different types of membrane proteins. In general, the proteins can be categorized into two types. Okay, you have integral proteins and peripheral proteins. So integral proteins, they're the ones that penetrate the hydrophobic interior. So they pierce through essentially the whole cell membrane. The majority of these are called transmembrane proteins. So they cross the membranes, as you can see here. Okay, the whole size of the protein, you have a little bit extending to the extracellular side and a little bit extending to the cytoplasmic side. And then you have, of course, it pierces through. That's the hydrophobic region. Okay, so it crosses the membrane. So this contains hydrophobic regions. We're talking about the protein. Remember when we talked about the different amino acids, and as you assemble and you undergo the tertiary structure, you have these hydrophobic regions of nonpolar amino acids that they can vary in length, but ultimately that's the area that will lodge into the hydrophobic area of the bilayer. Okay, usually alpha helices, right? Or the alpha helix, as you can see here. There's a bunch of alpha helices that are going throughout the hydrophobic region of the cell membrane. Okay, you have the hydrophilic regions. They're the ones that are going to be exposed of the protein into the intracellular, extracellular areas. Okay, generally these would create hydrophilic channels. Okay, where you can get hydrophilic substances to pass through the membrane via these proteins. For example, water. Okay, integral proteins, and then you have peripheral proteins, and these are proteins that they don't go through the membrane. They just stay at one side, so you really can't see it here, but let's say you can have a little protein that is just embedded right there. Okay, rather loosely, they're embedded, held onto by the cytoskeleton or whatnot, but they usually face one side of the cell membrane. Okay, so on the cytoplasmic side, you could have them, as I mentioned already, you can have proteins that are anchored, by attaching to the cytoskeleton, and then you can have others that are on the extracellular side, right? They're attached to materials outside of the cell. They have fibers, integrins, and they can work along with these structures to give strong framework for the cell. Okay, so you have integral proteins and peripheral proteins. So now we're going to looking more specifically proteins, now, and we're going to see different types of proteins. We're going to look at the general functions that membrane proteins can have, and we're going to start with very important panel to study. Right? You can see here, these are transmembrane proteins that they span through the membrane, and they can be channels, as you see here, right, where a molecule can essentially diffuse, right, or some solute can I don't want to say dissolve, diffuse through the extracellular side to the intracellular and vice versa. And you can have another type, carrier proteins, where you can have molecules being shuttled, okay, from one area to the other. It is possible to use energy, okay, to make it move from one place to the next, but it doesn't necessarily have to. You can have a type of passive transport, as we're going to see later. Okay, and the way that they shuttle the molecules is by changing the shape, okay, of the overall protein. And we're going to see this in a little bit. Okay, letter B, you have enzymatic activity. You have a protein that is, or a complex that's built into the membrane, and it could have enzymes that have active sites, and these active sites can come and bind with reactants. These reactants will break apart, and the products of that first reaction can jump into a second enzyme that will further break down into another product. Okay, so it doesn't have to be a complex. It could be, you can have solo enzymes or you can have them in a complex. Okay, and this, you often see these in metabolic pathways embedded within the membrane. Okay, you have signal transduction, where you have a special type of protein, which is a receptor, and these receptors can bind to very specific signaling molecules or ligands, right? An example of this could be a hormone, right? And this signaling molecule can cause the protein to change shape, and that change shape will relay a message to the inside of the cell, to another molecule, which can go further down into the cell. It can even go into the nucleus to initiate gene activation. Okay, so or transcription, right, to turn on a gene. So depending on the signal that the body is sending, right, or the cell is receiving. So signal transduction. You have cell recognition, so where some proteins have a carbohydrate chains bound to them. They're referred to as glycoproteins, and these serve as identification tags that are very, very important to recognize, who are your neighbors, right? What cells are you neighboring, right? And this type of cell binding is usually short-lived in comparison to what we talked about with the junctions, if you remember, with the parts of the cell. But this is more over for ID tagging. Okay, very important for embryonic development. Okay, intracellular joining, as I mentioned before, here you can have the different types of junctions, the tight junctions, right? That can hold the two cells together. Okay. And attachments to the cytoskeleton or the extracellular matrix, right, where they help maintain the shape, stabilize the cell in certain areas, stabilize certain transmembrane proteins to not move around so much. Okay, so you can have a coordination of extracellular and intracellular changes. So these are roughly the major groups. And let me just zoom out here. Understanding cell surface proteins, very, very important for medical science, for medical research, right? A particular case, right? You have a protein called CD4, and this protein is usually found in the surface of immune cells. Okay, and the surface of these immune cells helps the HIV, the human immunodeficiency virus, to infect these types of cells. So this CD4 gets recognized by HIV. Okay, and what happens? I'm sure we're familiar with HIV. This HIV virus ultimately leads to AIDS, right? To acquired immune deficiency syndrome. But interestingly, some individuals can get multiple exposures to HIV and not develop AIDS, or they don't show evidence of HIV infected cells, because these studies had shown that infected individuals actually require or had this virus binding to CD4. Okay, but also they had another co-receptor that they needed to bind to, which is CCR5. Okay, so gene analysis between these individuals and infected individuals had a variant of a cell surface protein called CCR5, and it was a co-receptor for HIV to infect most cells. Okay, so as you can see here in this little panel, HIV can infect a cell that has CCR5 along with this CD receptor, but HIV cannot infect a cell lacking CCR5 on its surface. So these individuals have a natural resistance, if I can say, to HIV. Okay. Ultimately prevents viral entry to the cell. And this is important because if you can find a way for drug treatments, this could serve as a way maybe to block CCR5. So those individuals that have it, maybe HIV can't use it, right? But it's quite difficult to do, drug discovery and drug treatments because any molecule that you're working with, there could be side effects, right? So interfering with CD4 has dangerous side effects. CCR5 is a safer target, and you have this Maraviroc drug that has been made. Okay, so it's just by understanding the idea of protein membranes can help in medical science. Good area of investigation. Okay, let's go to the next one. Okay. So the role of membrane carbohydrates in cell recognition. Okay. I just talked a little bit about this, but going into more detail, you want to be able to distinguish a neighboring cell from another. It enables you to have more efficient functioning of the organism. It enables you to create tissues, right? Tissues working together, cells working together. This is very important in embryonic development. As the organism is developing from a zygote, you need to have a distribution of cells, and cells need to know who's next to them so you can get a sense of the tissue types that you're starting to create. So very important for embryonic development. It's also important for the immune system when you want cells to target foreign cells, to destroy or reject foreign cells. You need to distinguish a friendly versus a bad guy, right? In a cell type, in different cell types. Okay, and the way that they do that is by binding to molecules. Usually, these molecules contain carbohydrates on the extracellular surface of the membrane. So these membrane carbohydrates, they're usually short branch chains. Okay, 15 sugar units, and you can have several types of molecules. We've talked moreover of glycoproteins, but you could also have glycolipids, where you have the carbohydrates bonded to lipids or bonded to proteins. So these carbohydrates that are on the extracellular side of the cell membrane can vary from species, can vary from cells within the same organism, right? So there's a lot of variety in terms of recognizing and ID tagging. So the diversity of molecules and their locations enables such a variety that they can serve as markers. For example, human blood types, they have variants of carbohydrates, right, on the red blood surface that is unique to red blood cells, and that can help medical treatment or diagnosis, right, when you do certain analysis, medical analysis there. Okay. This is a nice schematic of the synthesis of membrane components and their orientation. Orientation is always important because you need to know where you're going to add that recognition site and those recognition molecules to be used effectively, right? So just to go into this little notes here. Okay, synthesis and sidedness of membranes. So membranes have distinct inside and outside faces. Lipid bilayers may differ in their composition, one of lipids and how the protein is directed or has its orientation. Okay. So the asymmetrical arrangement of proteins, the lipids and their associated carbohydrates, can determine what membrane is being built and what it's going to be used for. Okay, So here's a nice schematic of the membrane components, right? So you have secretory proteins, membrane proteins, all coming from the ER. And there's a nice kind of schematic here where you have the inner layer of the lipid bilayer being in blue and the outer portion being in red for this particular structure. But then ultimately, when you see here, right, you have the cytoplasmic face having altered. All right, so that's quite interesting, right? Carbohydrates are added to the transmembrane from the ER, right? To the transmembrane, making them glycoproteins. So these carbohydrate portions can then be modified. Materials are transported in vesicles to the Golgi apparatus. So you have these particular molecules, and you have these particular vesicles where you have the attached carbohydrate on the inside, okay? And you have these secretory proteins on the inside. And as they're going along in the Golgi apparatus, they undergo certain modifications, okay? Let me just zoom in there. And you see these little modifications, and went from a Y to this little kind of branch conformation there, right? Where you're getting more carbohydrates, or you're creating glycolipids. And then, again, the vesicle is sent out, and you have these secretory vesicles that contain these particular molecules, and they're shuttled all the way to the outside of the membrane, where you now have a fusion of the vesicle and cell membrane, and the receptors and the ID tags have been embedded now into the cell membrane. So here's your transmembrane glycoprotein, here's a membrane glycolipid, right, that could be exposed to the surface of the cell for whatever it needs to be used for. Okay, so you're starting, from the inner, and you get out into the outer side of the membrane. Okay, so synthesis and sidedness of membranes. Okay, and the last thing for this video is membrane structure results in selective permeability, right? So cell membranes, of course, they have emergent properties. You have so many molecules that are embedded within the cell membrane, and that provides a very unique ability of the cell membrane is to be selective. Okay, this is referred to as selective permeability, allowing some substances to cross more easily than others, and this is quintessential, if I can say, critical to the cell's existence. Okay, and again, how is this possible? Is because of the fluid mosaic model. It enables the cells to regulate just because of how it's built, just because of the dynamics of the phospholipid bilayer and the proteins that you can embed within this membrane. Okay, so the form fits the function. Okay, as we've seen before, material can move across the plasma membrane in both directions. This is occurring all the time, right? Constantly, immediately. There's a chemical exchange between a muscle cell and extracellular fluid, just to give you an example. Sugars, amino acids, nutrients need to be entering the muscle cell immediately, because these are cells that are in constant motion, they're in constant work, we're moving all the time. Metabolic waste also need to leave immediate, right? Carbon dioxide, for example. So oxygen and carbon dioxide exchange must occur really relatively fast. You have ions, right, that need to be regulated quickly in the phospholipid bilayer. So there's a constant need of material coming in and out incredibly efficiently, effectively, quickly, right? And it's done because of the permeability, the selective permeability of the cell membrane. Okay. Generally, nonpolar molecules, okay, nonpolar meaning that they have no net charge, right, in different areas. They can pass through the membrane pretty easily because it's hydrophobic. The tails are hydrophobic, so they can just diffuse through the membrane. Hydrophilic molecules like ions and polar molecules, they'll diffuse rather slowly, if at all, through the bilayer, even water, right, compared to nonpolar ones. So in order for, molecules that you still require, but that they're polar, and ions that you require, you need transport proteins. Okay, so this kind of gatekeeping system enables this idea of selective permeability. Okay, again, we have proteins that are built inside the membrane that helps with this movement, thus the transport proteins. So they can't, polar molecules, ions, they can't move on their own. So they need these types of proteins. So you have channel proteins and you have carrier proteins. Okay. And channel proteins, the name tells you, it's like a little tunnel, right? And a famous one that you should memorize is aquaporins. So this facilitates water passing through instantaneously, right? Very, very quickly. And here is an example of how aquaporins would look like. What you see here is the cell membrane. Okay? In yellow, you have the head, the green tails, okay? The bilayer. So the aquaporin will be these alpha helices that you see here. It's a little bit difficult to see. All right. So this region and this whole protein in itself enables water to pass through a lot more effectively. So this all these little molecules you see here is water in red with white. Okay. But if you notice, there's no water in between the tails, but they do go through for these transport proteins. Okay. So just to go back to the notes here. So aquaporins, again, it's a protein, has 4 identical polypeptide. Sorry, I kind of repeated the word there. Polypeptide subunits. They form a whole tunnel, and that tunnel would enable water to pass in a single file. Okay. 38 molecules. Sorry, 3 billion molecules. I saw that wrong. 3 billion molecules of water pass per second. That's how effective this these proteins can be. So you have carrier proteins. So moving on to the next protein, you have carrier proteins. They hold on to their passengers. Again, like I mentioned before, we're going to see these in more detail in the later sections. But they hold on to their passengers. They change shape, these proteins, and they change shape in such a way that they get shuttled and pass through across the membrane. Very, very specific between the substrate, the molecule that is binding to the protein, and the protein. Okay. For example, glucose carrier proteins. Red blood cells, right? Glucose carrier proteins. Glucose moves 50,000 times faster than on its own. Proteins are so specific that glucose can bind to this particular carrier protein, but fructose cannot. And fructose is just an isomer having the same chemical formula but a different structure, cannot bind. That's how specific proteins are. Okay, so ultimately, selective permeability, right, will enable specificity, a very tightly regulated movement of material. Okay, the membrane depends on both discriminating barrier of the lipid bilayer and the specific transport of proteins, of specific proteins. So very, very interesting how the membrane works. Okay, so that will finish the first part of chapter 7. I'll continue chapter the second part, 7b, in soon. Okay, so thank you for listening, guys, and I'll see you in the next video. Bye bye.